I'm going to deviate from the program for a moment. I've had a few questions. There's a few little tidbits that you might want to carry away. Okay. This is a web page that is worth knowing about. Anyone using flexible containment in laboratories, they have a wonderful web page. They've been making flexible barriers for 50 years that I've been using them. And $15, throw away 2 mil thick polyethylene. Very useful for weighing powders in laboratories. You can throw them away, no problem. And it's a good way to get some practice. Plus, they have a beautiful little hints and tips book. You used to have to wait 2 years while they published them. Now it's on the web page. You just pick it up, and it's got wonderful ideas on how to apply containment as a general concept. And of course, it's built around their polyethylene glove bags. We've also talked about visible limits of dusts. You can enhance your visible limit of dusts. Julian.
There's a slight problem. Some of us have bad eyesight.
I'll leave it up here. It's i-2-r.com. I2R, you may know it as Instruments for Research and Industry. They've been around for many, many years. It's a small mom-and-pop operation, and they make a unique range of products that some of them you might find useful beyond the containment side. Laser pointers. We talked about visibility of dusts, but our toxicology people who did inhalation studies told me this. If you use one of the old type laser pointers that have the ability to produce a wide beam for underlining, you can see dust at much lower concentrations than you can with normal visual acuity because you watch the light scattering, the Tyndall effect, and you can go down to about 100 times below your normal visual acuity using a laser beam. So you can enhance your smoke studies even using this because you can see smoke trailing away 25 feet beyond where you see it with your naked eye. Also, in conjunction with these, when you've got this bag full of air and you now want to put it in a waste drum, it's kind of like fighting an octopus. So if you don't put a relief HEPA filter on it when you start, you're not going to get it in the bag. So we use a North respirator cartridge. It's the only remaining respirator cartridge that has a screw fitting on it where you can secure it in the glove bag. And we use a piece of duct tape on the outside of the glove bag, a piece of duct tape on the inside, cut an X in it, push the threaded part through the X so that it kind of forms a gasket, screw it on the inside, and now you have a collapsible device. Plus, it will also take the stress of the operator putting their arms in and out of the containment. ZL5, I wish we'd had it when we did our study. ZL5 is a material we came across 2 years ago when we were doing some cleaning studies. It's a beautiful material for training operators with. It's much more sensitive visually than riboflavin or fluorescein. It's detectable in solution down to 40 parts per billion with a UV light. It looks like ethylene glycol visually. It's a rust-colored powder, but when you disperse it, you can't see it. The minute you hit it with water, it turns brilliant yellow, so you can train the operators how to clean. And then when the yellow's disappeared, you take the UV lamp and you follow their footsteps across the floor because the green glow is detectable down to 40 parts per billion. The one atom, one molecule issue that's coming up in the regulations, please shoot it out of the sky. Scientifically, totally stupid. Avogadro's number is very large, and even with our the best measurements technology today, we can only measure down to 10 to the 9 molecules per cubic meter of air. Okay? So we're a long, long way away from any one molecule theory. We went through this with acrylonitrile in the States back in the 1970s. We had to shoot the stupidity into the regulations. I was also asked about analytical detection limit for testing. Lactose is very nice. It has no vapor pressure. Naproxen likewise. This is the biggest problem you're going to have. You should be able to get down to 10 parts per trillion with current technology for most materials. But you may airstrip the material off your filter during sampling if it has a vapor pressure greater than 10 parts per trillion atmospheres at room temperature. And many of your compounds do. Okay? So that's a practical limitation to actual sampling. Now we'll get back to the agenda.